Here's the primary structure of the stems, or the stem cross-section of the primary tissue of a plant. So this is the tissue arrangement of monocots versus eudicots. In general, we'll go over each of these, but I'm just standing here for a second, and you can stop the video and do some drawings. All right, so let's kind of move in a little bit closer. So in the monocots, the main kind of arrangement is scattered vascular bundles within the C of cortex. So all of this tissue in between the vascular bundles is called cortex. It's basically made up of parenchyma. And of course on the very outside would be the epiderm, including cuticle and stomata. Most stems are photosynthetic, so you would see the actual cells of the parenchyma or cortex towards the outside being very, very green and photosynthetic. And as you get more internal in the plant, of course, it's away from the sun, so there's less chloroplast and less photosynthetic. In terms of the eudicot, let's just look at that. The eudicot also has the epiderm and cuticle, including the my dog is like squeaking, sorry. And then the cortex on the outside of that. But a different kind of arrangement here in that there's a vascular cylinder with vascular bundles arranged in a ring with an area of ground tissue in the middle called pith. You might remember that the monocot root had pith, but the monocot stem now does not. And the eudicot root did not have pith, but the eudicot stem does. So I just kind of point that out. Also, and some other features of this drawing here are the xylem is always towards the middle. By the way, in the monocot, that is also true. And the phloem is towards the outside, so the phloem shown here in green. And then the pith, where it kind of extends towards the vascular cambium, makes sometimes a little more elongate area here called the pith ray. On the other side, you could also call it a cortical ray, although they don't usually point that out for some reason. Not really sure why. All right, so let's look at a single vascular bundle of a monocot. You're going to have a bundle sheath cells, which include fibers, or maybe just mostly fibers, sclerenchyma cells. And then you have xylem, which is going to be towards the inside, and phloem. And in the monocot, there is a layer of vascular cambium that's laying down new xylem and phloem as the actual bundle develops. But then after the bundle is completely developed, it will no longer be present. So this is what we call a closed vascular cambium. It does not extend towards the other vascular bundles within the stem. Now the vascular, so looking at a vascular bundle of a eudicot, we have, this is towards the inside and this is towards the outside. You have the same kind of arrangement in the terms of the xylem is towards the inside, the phloem towards the outside, and vascular cambium in between. But in this case, it extends between different vascular bundles. So right here it's shown in red. So basically it's kind of a continuous layer. And in the eudicot, it's also going to act for the secondary growth, laying down new xylem towards the inside and new phloem towards the outside, like we talked about in the root, but we will talk about again here. And because the vascular cambium can exist both in the vascular bundle itself and in between the vascular bundles, they have different names. The intervesicular cambium on the outside, that's the extension one between bundles, that's inter, and vesicular cambium would be within. Sometimes they call it infravesicular instead of inter. So anyway, those are just some terms. And the other thing that's a little bit different is there's usually a bundle cap, like a whole group of fibers that is on the end towards the outside of the stem. This adds support to the stem because generally speaking, because of the cylinder arrangement, you need a little bit more vertical support of the stem to hold the leaves up than you do in the monocot. Now the monocot has, because it has scattered vascular bundles, and when you see this actually in reality, it's a whole lot of vascular bundles, not just like sort of random like I have drawn here. If you think of a palm tree and if you cut it off and all those little things that are poking up, those are all the vascular bundles of the palm tree. So there's a lot of them. So they don't have the bundle cap scenario, generally speaking. Okay, so, and I think that that's generally the plan. Just to kind of give you another view, this is sort of the vascular, this is another way of drawing that. So sometimes they look a little pointy towards the middle. Whoops, this is backwards. It could also look a little more continuous in some plants as opposed to having a distance between them, but still always in the eudicote, the pith in the middle. Now the pith is ground tissue, generally speaking not very photosynthetic because it's very internal. All right, so let's look at a model of this. I'm gonna move it down here so you can see it. This is just a model showing you more of the kind of different kind of colors here. This is just showing you the epiderm. It may also show you, I'm not sure here, they're trying to show you stomata here. It's not very clear, but anyway, the epiderm may have stomata. It might also have trichomes too, depending on the stem type. This is just showing you the layer of cortex that has high photosynthetic value, or that is, it's very, very green because there'd be a lot of photosynthesis happening there. And then as you get more internal, a little less so. This is the vascular bundle, the bundle cap, the phloem, the vascular cambium, and the xylem. And then this is the pith in the middle here, and this would be a pith ray. So just kind of pointing it out on the 3D version. Sometimes the pith of a plant looks a little bit like hollow, and you might have seen that before. And this is just showing you the vessels from the side. You might remember that the vessel members and tracheids have on the side of them pits. It's not really showing you the annular rings per se very well. A little bit on this one. You can see the annular rings on that. And this, of course, is vascular cambium, so it's undifferentiated parenchyma, and that is doing mitosis for xylem and phloem. And this, of course, is the sieve tube elements and companion cells, so it's kind of nice. You get to see them from the side. And these are mostly fibers. Remember, fibers are sclerenchyma and dead. These are cortical parenchyma cells, and then, of course, the epiderm.